Good morning, everybody, and good afternoon if you're overseas. We want to welcome you to this symposium on the new science of skin barrier damage and repair and its implications for childhood atopic dermatitis, or eczema. I'm Jeff McBride, dermatologist, dermatopathologist, and cell biologist from the University of Oklahoma, and I'm a board member of Global Parents for Eczema Research, or better known as GPER. For those of you who aren't already familiar with this organization, it's a global nonprofit founded by parents and focused on improving the future for children with moderate to severe eczema. This annual symposium spotlights cutting-edge research that holds the promise to improve the lives of children and families impacted by this condition. And we are thrilled to host today's lineup of speakers. We'll be talking today about the skin, the body's largest organ, and a critical barrier meant to serve as a defense against harms from the outside world, as well as a sealant for the body that maintains essential hydration for the skin. We're going to learn today about what happens when the integrity of the skin barrier becomes damaged through a variety of assaults, including chemical exposures. Once it is damaged, this leaky skin barrier can no longer serve its role to prevent allergens and microbes from penetrating into the body, which then leads to a runaway inflammatory response, as we see with conditions like atopic dermatitis. This symposium is more than just a meeting. It is a platform for collaboration, learning, and the exchange of ideas. To that end, we invite you to pose questions through the webinar, Q&A, or by raising your hand if you're here in the room. We'll include time for Q&A after every presentation. We'd also like to take a moment to thank our sponsors of today's event, Pfizer, Pierre Fabre Foundation, and Kinview, as well as members of GPER's Corporate Council. Without further ado, I will now pass the mic to Corey Capozza, GPER's Executive Director, who will introduce our first speaker. Thank you.
Thank you, Jeff. Good morning, everybody. It's a pleasure to be here today. I'm Corey Capozza, Executive Director of Global Parents for Eczema Research, and I'm also the mom of a child with moderate to severe eczema, so this topic is of personal and professional interest to me today. And it's really my pleasure to be here and to introduce our roster of speakers. Our first speaker is Dr. Lisa Beck. She's co-director of the Center for Allergic Disease Research at University of Rochester Medical Center in New York. For 25 years, she has dedicated her career to studying this condition, to finding safe and effective treatments for patients, and understanding the skin barrier changes that occur with atopic dermatitis. And today she will be talking about how type 2 inflammation impacts the skin barrier with atopic dermatitis. So welcome, Dr. Beck, and thank you for being with us.
Thanks very much. And I wish I could be sitting next to you. Trust me, Rochester, New York does not look like California right now. So you're going to hear a lot of overlapping themes in today's speakers, but what I'm going to talk to you about is barrier, and specifically the role that type 2 inflammation may play in this barrier abnormality that we think is characteristic of this disease. So like all of the speakers, I need to let you know that I consult for a number of companies developing drugs for atopic dermatitis and chronic urticaria. And I have been an investigator for Dupixent trials for atopic dermatitis. When I talk about atopic dermatitis, I always think of this as a disease for which there are 4 key features. And any discussion about pathophysiology would need to take into account explaining these. And they are the type 2 inflammation, which we think really dominates in most eczema patients of all ages, even though there can be some other pathways. And that's really saying that we have a strong influence of 3 different mediators or proteins, interleukin 4, 5, and 13. We think that microbial colonization also is a key feature of this disease. And what's quite interesting is this colonization state is found not just in the lesional skin, but also quite commonly in the non-lesional or apparently normal looking skin. And then, of course, we need to talk about barrier, and we're specifically for this talk talking about the skin epithelial barrier and itch, which we think is really 24/7. This cartoon is meant to also show that the arrows go in every direction, which really means that these all seem to influence each other. And I think we need to look no further than the moniker that was coined over 150 years ago, that eczema is the itch that rashes. And so we know that underlying inflammation is what's driving the itch, but if you respond to that itch with scratching, then you will get barrier dysfunction and then more allergens get in. So that's just kind of a very superficial view of how all of those four pathways really dynamically interact. And we think eventually we'll understand all the mediators that explain that. So atopic derm is characterized, particularly in adults, with lesions occurring in particular anatomic areas shown here. Although when the people are bad, it can affect, 80% of the body surface area. We also recognize that for most people, there are areas that look pretty normal. The skin may be a bit dry, but there's no redness, excoriation, thickening, or bleeding. But it turns out that this non-lesional skin is really not normal. It is really abnormal. It has barrier defects. It has staph colonization. It sometimes has a small component of itch. And when you biopsy, there is inflammation, just not as much as we see in lesional skin. So I think one of the things when I talk about barrier is important to note is it's remarkable how well it works for most of us, the skin epithelium. And so here's a histology of this multi-layered skin epithelium, starting from the basement membrane zone down here and going all the way up to this area we call the stratum corneum. And a very orderly differentiation occurs during that time, and it can take about 14 days to go from this cell to the top. So it's always rapidly turning over. And when it works well, it really protects us from seeing any one of these common environmental allergens as something foreign and responding to them. So we're obviously all exposed to common allergens, but only a few of us really immunologically respond to them adversely. We're also exposed to, millions of microbes, bacteria, viruses, and fungi, and yet most of the time we don't respond with inflammation or barrier disruption from those. And more increasingly, we're thinking about pollutants and climate change and the effect that has on the epithelium. We also put our skin in wet environments and dry environments and in hot and cold environments. And yet, for most of us, it provides that critical barrier structure. So I just want you to thank your epithelium right now, because for most of you, it's doing a really good job, but for eczema patients, it does not. So for the purpose of this talk, we are going to focus on the cartoon here and look really at the epidermis part of this, not the subepidermal area called the dermis. And we'll highlight some of the key factors that are abnormal and probably promoting this barrier defect. And then we'll start with the outer area first, which is what we call the stratum corneum, where we get epidermal cells that flatten out like pancakes. And how well they flatten out is determined a bit by the amount of filaggrin they express and the cross-linking that filaggrin does to other proteins. And that compacts that epidermal cell into the pancake. That's part of the barrier structure for stratum corneum. But the other part is what bathes those corneocytes or stratum corneum cells, and that's the lipids. And they are a very complex area of human biology now, because it's not just the relative expression of different lipid moieties, but it's how they are laid down in a 3D arrangement. And both the lipid composition and how they are laid down is abnormal in atopic derm patients. So we think that we lack the ability to form these really compact corneocytes, and surrounding those corneocytes, we have abnormal lipid structure. For reasons we are still not sure, we also don't develop the very acidic pH that is typical of normal people's epidermis. And so in atopic derm patients, it's more alkaline. And then once you move below this first outer layer that's very good at protecting us as a barrier structure, there's a barrier structure called tight junctions that are present just below that, right along this area. And we know from atopic derm studies that they are also dysfunctional, as they are in the airway for asthmatics. So the consequence of these barrier abnormalities is that we can sometimes use a device that's non-invasive to measure what we call transepidermal water loss. That's how much water is coming from the inside to the outside. So in that direction, and we use the abbreviation TOOL for that. And so if our patients have quite a bit of barrier disruption, they will have high transepidermal water loss. And those will be our patients with drier skin. But we know when the barrier is disrupted, that also allows the pathogen that I mentioned to you is commonly colonizing the skin of AD patients, namely Staph aureus, that can gain access to the immune system and elicit a lot of inflammatory cascades and even activate the T cells and drive a T cell derived inflammation. We see greater penetrations of allergens, irritants, and microbes. We see greater production of proteins by the epidermal cells when they're disrupted, of proteins like TSLP that promote more type 2 inflammation. And so this is sort of a master type 2 driver, and it's commonly produced by epithelial cells when they are disturbed and the barrier is disrupted. And lastly, an area of more recent interest is when barrier is disrupted, these antigen-presenting cells that we call Langerhans cells that are present in the epidermis, their dendrites now start coursing up into close to the surface of the skin so that they can now respond to things that we don't want the skin responding to. So we like them when they stay low and only respond if there's a lot of epidermal damage. And the same is true for the sensory nerve endings. They now can be triggered by a number of allergens that are proteases and trigger an itch response. So you're going to hear more about this interesting study done to try to identify whether whether there are any features that you can identify from tape strip skin, STS, at a very early time point in life, at 2 months of age, that would help you predict the children that at 2 years of age develop atopic dermatitis. And so the study took the majority of the patients from a high-risk group, meaning both parents had atopic conditions, or those with no risk, meaning both parents had no atopic conditions, did this tape stripping, measured a variety of lipids and mediators from the tape strip, and asked what helped them predict those that went on to develop AD. And you can see that in the high-risk group, almost 30% developed atopic derm at 2 years, and about 13% of the low-risk developed atopic derm. And this is what they found. So they found that family history helped to predict it. And so we already knew that's a twofold increase odds ratio. If they had inflammatory markers that mark type 2 polarization or type 2 inflammation, namely that master switch protein TSLP, or a type 2 cytokine itself, IL-13, that having any of those significantly increase the odds ratio that an infant that you detect these at 2 months will develop AD at 2 years. But then if you add to those measures of the barrier abnormality, specifically they were focusing on lipid moieties, now you remarkably increase the predictability of AD. So it's kind of combining a type 2 marker and a barrier marker that really gets you this increased insight of what's the risk for that child developing atopic derm. So for this quick talk, it would be impossible to tell you what are all the pathways by which these type 2 cytokines, IL-4 and IL-13, might mediate barrier disruption. So this is a summary slide giving you big categories of what type 2 inflammation does that might be problematic for the barrier. So it can affect the lipids, increasing short-chain fatty acids and decreasing long-chain fatty acids. And we think that leads to these barrier abnormalities. It can affect a number of other structural epidermal proteins, largely decreasing them. And they too, when they are decreased, would lead to physiologic measurements of barrier dysfunction. We know that the epidermis of AD patients have more protease production, and we can observe in the test tube that type 2 cytokines can lead to that, as well as reduced expression of proteins important for that second barrier structure, namely the tight junctions found right below the stratum corneum. They too are affected by type 2 inflammation. And this decreased tight junction function isn't just in the epidermis, it's all the way down into the sweat glands and sweat ducts, which is why we think some of our patients tell us that they itch when they sweat. And it turns out it may be because their ducts are so leaky, the sweat leaks out into the skin rather than getting to the skin surface. And that may also be why they're more susceptible to heat exhaustion. And there are a number of other effects they have on the epithelium that would help promote staph colonization. So you all know this new biologic, the first biologic approved to treat atopic dermatitis, and now approved to treat a number of other allergic comorbidities shown on this slide. And this biologic antibody is unique in that it binds to a subunit of the type 1 and type 2 receptor, namely the IL-4 receptor alpha. And by doing so, it blocks the ability of IL-4 to bind to both receptors, because IL-4 can mediate its biology by acting through both receptors. But IL-13 primarily mediates its biology only through the type 2. So by targeting this receptor subunit, it has effectively blocked both 4 and 13. So we recently completed and published a NIH and Sanofi Regeneron funded study where we were looking at more mechanistic questions, trying to ask if you use something that is highly effective at blocking IL-4 and IL-13, what effects did we see on barrier and on staph colonization? And so let me just highlight that this was a typical randomized double-blind trial where patients 2-to-1 were randomized to dupilumab or placebo. These were all adults with moderate to severe disease. And during the first 42 days or 6 weeks, it was randomized. And then to encourage patients to participate, we had a 10-week open label so that all the placebo patients would know that they would at the very least, get 10 weeks of treatment. And probably the result that was most shocking to us was that we saw this unbelievably dramatic drop in the dupilumab assigned group in Staph aureus, both in their lesional skin and non-lesional skin, that was not seen in the placebo group, but was seen when the placebo population was allowed to have open label dupilumab. And 5 weeks after they were given that dupilumab, they too saw this dramatic drop in Staph aureus. So again, by day 3, we already saw a separation. What we thought was interesting was this is sort of the timeline of the significant changes observed in this 6-week period. And again, in the first 3 days, as I showed you over here, the Staph aureus was reduced non-lesionally and lesionally. By day 7, we could detect reductions in type 2 mediators, both in the blood and in the tissue. And it wasn't until day 14 that we saw a separation where the Dupi-treated group was statistically better by all metrics of clinical improvement compared to the placebo group. But it took till about 6 weeks or 42 days to see that the barrier improvement was seen. So this is a later improvement from type 2 blockade, but maybe an improvement that helps to explain why when patients, for whatever reason, go off of dupilumab, we tend not to see their disease return to its previous baseline, typically for weeks and sometimes even months. And so that measurement, again, was using this transepidermal water loss tool. So we know that dupilumab, another group, did a similar study. This was not controlled, and it was adolescents and adults. And in this case, their comparison was back to the patient's baseline. What they were looking at were some of those lipid abnormalities that I told you are abnormal in atopic derm patients. And this is sort of a cartoon of their results, where those lipids that are overexpressed in AD now became reduced with dupilumab treatment, and those that are reduced in AD compared to controls started to normalize and increase. So we can see some readouts that are not functional, but are characterizing barrier features normalizing. Importantly, if we look at filaggrin, one of the structural proteins found in the epidermis, you can see that before treating with dupilumab, if you look in green, this is the filaggrin expression that's in the upper layers of the skin. And after therapy, much more green, so much higher expression. And if you quantify that, you can see that there's an increased expression after therapy, and it almost equilibrates to what you see in normal non-atopic patients. So in conclusions, I wanted to just highlight that barrier defects are a cardinal feature of this disease. They're more profound in lesional skin than non-lesional, but they are abnormal in both. And that the skin barrier defect is thought to be responsible for a number of the key features we see in this disease: dry skin or xerosis, staph colonization, infections, allergen sensitization, promoting more type 2 inflammation, and itch. And some of these specific barrier abnormalities may be due directly or indirectly to the actions of the canonical type 2 cytokines, IL-4 and IL-13. And therefore, we can be hopeful that by blocking those or reducing the biology of those cytokines either together or 13 alone, we'll see some improvement in barrier function. And I just wanted to end just to highlight that I'm always grateful for the groups that have funded this work and the patients that allow us to study them, and a really talented group of people back at the University of Rochester and the NIAID funded network called Atopic Dermatitis Research Network. Thanks very much.
Thank you so much, Dr. Beck, for that excellent presentation and quick tour around some very complex research and topics. I personally learned a lot in your short presentation. And one of the things that was particularly interesting to me is I always wondered why my child with atopic dermatitis both seemed to sweat less than other children, but would also experience sweat as a trigger. And so your explanation, that was the first time I heard that before. It's very interesting. One thing that I found especially interesting about your presentation is often when we think about Staph aureus overcolonization in individuals with atopic dermatitis, we might think the best strategy to address that would be through an antimicrobial or an antibiotic. But you found that you were actually able to see a large decrease in Staph aureus simply by addressing inflammation, type 2 inflammation, using dupilumab, and that reduction sort of preceded the clinical improvements that you later saw. So I wanted to ask that, given that you saw the Staph aureus reduction on the skin preceded the clinical improvement after use of dupilumab or this biologic, does that suggest that alteration of the skin microbiome is necessary for resolution of atopic dermatitis symptoms?
That's a really great question. And I think I want to start first by just highlighting for the group that there have been Cochrane reviews on various approaches to reduce Staph aureus in atopic derm patients, asking the question is, does that result in enough clinical improvement? Now, those were done all before dupilumab and tralokinumab, both of which have data showing that they reduce tralokinumab's anti-IL-13, showing that they reduce Staph aureus. But the upshot of the Cochrane review was that there isn't clear evidence that an anti-staph approach works. But keep in mind, most of those were pretty global, using antibacterial agents that would broadly kill many bacteria, not just staph. So the feeling a little bit about why we haven't seen a clear improvement with, let's say cephalosporin treatment systemically of our patients, or a tetracycline for those that we can give tetracyclines. We will see their staph decrease. The problem is we also see some good bacteria decrease as well. And we think that that might be part of the problem, is that we are not very targeted at reducing staph only. Dupilumab, on the other hand, does appear to be more targeted based on microbiome studies, meaning we're seeing a relative increase in other good bacteria and a specific decrease in Staph aureus. So it does, I would agree with your notion that it's possible that really good therapies for atopic dermatitis patients are going to have some positive effect on Staph aureus. Not because I'm not a believer that Staph aureus initiates this disease, although there is data that says that one of the risk factors that you can look at in infants to predict who will develop AD are those who have higher Staph aureus on their skin before AD develops. So there is a little bit of data that might refute what I said. I look at Staph aureus as an amplifier of the problem. And I think it's a little bit more a consequence of the underlying disease that you get this profound Staph aureus. We don't see it, for example, with psoriasis. So last year we studied that. And although we can find some Staph aureus on psoriasis patients, which is a really 17-driven inflammatory disease, there's no relationship of Staph aureus quantity and disease severity in psoriasis, whereas there's a very tight correlation, no matter how you measure Staph aureus, with disease severity. So I think it's going to be interesting to look at the JAK inhibitors and see what effect they might have on the skin Staph aureus. But I did want to highlight that 13 alone, at least based on a report with tralokinumab, suggests that it may have fairly similar effects to dupilumab. So you may not have to block 4 as well to get that drop. We're still trying to understand why you get that drop in Staph aureus.
Yes, and I think that's one of the exciting things about the research that you and others are doing is you're beginning to untangle that relationship, which is so typical of atopic dermatitis. I wanted to remind listeners that you can pose questions in the Q&A, and we will ask those in the room today. There is a Q&A at the bottom of your Zoom screen, and feel free to drop your questions in there for our speakers. I want to thank Dr. Beck. I know you have clinical duties right now. So again, thank you so much for joining us today, and we really appreciate your sharing of your knowledge with us.
Thank you, everyone.
Well, it's now my pleasure to introduce our next speaker, who is Dr. Chesmi Actis. He's a global leader in immunology specializing in allergic diseases. He is the editor-in-chief of the journal Allergy, director of the Swiss Institute of Allergy and Asthma Research, and a professor at University of Zurich Medical Faculty. Dr. Actis couldn't be with us live today, but recorded his presentation ahead of time, which we'll play now. And I really love his research. I think he is really pioneering new ways of understanding why we're seeing such an epidemic in allergic diseases. And he'll be talking about how chemicals can damage the skin barrier or the epithelial barrier in the body, which is this layer that protects skin, gastrointestinal system, and airways, and provides this protective wall against environmental factors. So with that, I'll play Dr. Actis's talk. One moment.
It's a pleasure to be here, Corey. The first allergic disease description was at the time of maybe Egypt. When we come to 1900, in Swiss Army, there was still 1% cases. And in 1960, again, it was 1 to 2% allergic patients. But after 1960, a very steep increase started. And at that time, atopic dermatitis, asthma, and allergic rhinitis started to increase. And they doubled or tripled within the first 10 years. Around 2000, there was a second wave of diseases. We started to see a peanut butter-induced anaphylaxis. Currently, food allergy is 5 to 9% in the world. So this increase is very, very steep. So what happened after 1960s to cause the global health crisis is one can understand it from certain chemical inventories. And there is 22 chemical inventories from 19 countries have been published. And it shows that 350,000 substances have been introduced to human lives with almost no control on their health effects after 1960s. This was a huge number. But many of them were not appropriately reported. 50,000 of them are publicly unknown because they are claimed to be patented, and 70,000 have been very ambiguously described. So we are talking about a major threat to humanity, like the global warming and climate change. In 1960s, we had the exposure to laundry and household cleaners and the detergents that are used for this. And this exposure to these detergents was followed with some certain surfactants, sodium lauryl sulfate, sodium dodecyl sulfate. They were introduced to our lives. Then we have diesel exhaust, which started to increase a lot at that time. That affects the respiratory area. And what affects in addition to the respiratory area is ozone increased and nanoparticles, microplastic, and particulate matter in the air. So air pollution increased, and substances that cause inflammation in the air increased. Then there is another group of substances that are affecting our skin: shampoos and body cleaners, household cleaners, surfactants started to increase and their usage increased, and we started to get exposed to them. And the third group is a change in our food. So we changed to the direction of packaged food, and this packaged food additives, enzymes, and emulsifiers in processed food are also responsible for some group of gastrointestinal inflammatory diseases or systemic diseases that are caused by gastrointestinal inflammation and damage of the epithelial barriers. So if we want to talk more about epithelial barriers, currently around 1/3 of the world population have a epithelial barrier problem. Their epithelial cells, which should not even pass water through, are leaky. And they are not so strong, and the epithelial cells cannot protect our body to environmental agents. Together with the environmental agents, let's say for skin, we are talking about now shampoos, household cleaners, body cleaners, again, air pollution that affects the skin, and allergens, toxins of the bacteria, pollutants, and the microbes of our body or the environment, like the opportunistic pathogens, can go deeper in these tissues. So skin and respiratory mucosa are both affected in this way, and they become inflammatory. So they start to respond to these agents together with their response to allergens. Then they start to recognize allergens as a dangerous substance. Before 1960s, we were tolerating allergens. Nowadays, we are not tolerating allergens. In the very beginning, the barriers are closed and they are opened by substances. But once they are open, when the inflammation starts and the disease starts, then they stay open so that they are exposed to environmental other substances like allergens, toxins, pollutants that makes the disease worse. Within these substances that we are being exposed, for example, if you take SDS, sodium dodecyl sulfate, which is also called sodium lauryl sulfate, if you put a few drops to the nose of mouse, eosinophilic, allergic, asthma-like inflammation starts. And we can see that a very similar mechanism starts alone. So this is only single exposure of a single substance. This gives us the impression that these substances are initiators of this response. But once this response starts, once inflammation starts to regulate the barriers, and inflammation keeps the barriers open. There's a nice example. In 1970s, in a production facility of a detergent, there was an accident, and the powder detergent has gone to the even offices of secretaries, and it had affected almost everyone in the facility, which was around 400 people working, and 380 of them developed asthma. And none of them had asthma before. But if you are exposed to a high dose, then you develop the disease. Currently, we have good evidence on occupational diseases like cleaners' eczema, cleaners' rhinitis, so that they are exposed to these substances in high doses, and cleaners, house cleaners, asthma. So these kind of diseases are existing and they are still increasing. So we are quite confident for the sequential events that are taking place in these patients. We were worrying around 2 years ago, what could be the reason for this second wave, which started to increase around 2000s? And we were thinking of several factors which were introduced to our lives for the allergic diseases that are coming from the inflammation of the gastrointestinal area. And we listed several substances that were introduced to our lives. And from these substances, professional dishwashers appeared as number one. We were using household dishwashers since 1980s. And household dishwashers may have an effect. But we then compared the household dishwashers with the professional dishwashers. Professional dishwashers bring out hundreds of very well cleaned and shining cutlery, plates, and glasses. So they are washing in 1 minute with 82 degrees temperature and 4 atmosphere pressure. In the last 20 seconds, the rinse aid is added, and rinse aid kills the foam And then at the end, the rinse aid is not rinsed itself, it stays. And it is the same for many rinse aids or softeners in laundry also, or household dishwashers. And the rinse aid stays on the glasses. So we were interested in this, and we tried to extract the rinse aid from the glasses. And we saw that a substance in the rinse aid called alcohol ethoxylates was highly toxic to human gastrointestinal epithelial cells, epithelial organoids, or gastrointestinal tissue made from stem cells. So whatever we tried, this rinse aid appeared to be toxic in the doses or in the dilutions of 20,000 times. So it's a very high dilution. And when we extracted the rinse aid from the glasses, and you put extracted rinse aid from the glasses to cultures and see that some cells that we are using in cultures are dying. Why would a dishwasher rinse aid kill our cells? That was quite problematic. Then we saw that when we make barrier systems, when we put the epithelial barriers into these cultures, it was breaking the barriers. We were measuring this very carefully. And then the barriers were broken. And then we understood that this alcohol ethoxylate is the main culprit. And it is it can be extracted as a remnant after professional dishwashing. And it also exists in household dishwashing sometimes as a part of the tablet. And so it appeared to be a toxic substance which was introduced to our lives in around 2000s. Currently, it is being used in hospitals, armies, in many restaurants. So it's something that the health authorities should really think about and focus. The amount of packaged food usage has very substantially increased. And there's emulsifiers, preservatives, surfactants, or additives of these food. And the commercial aim was to extend the shelf life and prevent the separation of the liquids of the packaged food. So this commercial aim has been successfully achieved. About 20 years ago, the shelves of big supermarkets were always changed every week because the shelf life of food was not longer than 7 days. Currently, shelf life of many food is 6 months to 1 year. So they stay in the shelves and they are not contaminated with fungi and bacteria. So there are additives to prevent the growth of fungi and bacteria. And in addition, there is some surfactants added or emulsifiers added to keep the water inside. So this is all achieved, but to what cost is it achieved is a very major question. Currently, we have a very long list of emulsifiers used in the USA and Europe. There's around 100 emulsifiers. And the concept of an emulsifier is to keep the water inside the food, and there's a thickening effect. And we are ready to publish a new paper, which is about some ice cream and pastry additives, polysorbates, and they are quite toxic. And the level of evidence when we think of food emulsifiers, we don't know directly the human toxicity, but mouse toxicity is very clear. There are so many papers that these emulsifiers make gut inflammation, open epithelial barriers, change microbiome in mouse. And if we come to microplastic, and microplastic, we have been introduced to microplastic about 20 years ago. 8 billion tons is produced per year plastic, and around 1 billion ton of microplastic goes to our lives. We have data about microplastic ingested by cells. It changes the immune responses. So it is everywhere nowadays, and it is one of the main also agent that we are continuously being exposed, that changes the cellular metabolism, cellular activation, and causes inflammation. And we think that this is exposure to toxic substances, and which ends up with the development of about 1/4 of the world sick. So what is happening is that it is a major change related to epithelial barrier damage, microbial dysbiosis, and immune system activation. So the first thing is avoidance and dose control. all of the above mentioned products. So we should decrease the exposure to below the toxic doses. That's technically possible for food emulsifiers, packaged food, very easy. For air pollution, it's difficult. For certain days, the air pollution, depending on where you live, is very high and it's not controllable. And then you have to have air cleaners in the house. If there's a patient, then air cleaners should be used a lot. Then one should think of synergistic and additive effects of these substances. You know, if there's house dust mite at home, and at the same time there's air pollution outside, and at the same time you are feeding your child with a packaged food which has emulsifiers, these substances have additive effects. And for the industry, there's a responsibility of development of safer or less toxic products. And the regulatory authorities should certainly control these. They would certainly say that these doses are still toxic, less should be used. Then we have to discover biomarkers for research. That's very important to identify barrier leaky subjects. We are working on this, and we have some devices to show it in the skin. It wasn't so easy in the beginning to bring these concepts to this level, but from now on, I feel that it is also like the avalanche of healing, not the avalanche of polluting, but the avalanche of healing is starting.
Thank you to Dr. Actis, who couldn't be here today, but a sweeping presentation that I think starts to get at that question of why are we seeing so many children developing these allergic diseases, including atopic dermatitis, and really pointing to these new exposures to chemical substances that did not exist in the past, and those exposures really coinciding with the increase that we're seeing. I would invite others in the room, if you'd like to make a comment or a question, please feel free to use the microphone. And we are seeing some cute questions come in the chat as well, which we'll try to answer. Because Dr. Actis couldn't be here today, we did pose a question to him ahead of time that came from parents. And the question was, given that we are now exposed to all these different types of toxic exposures, what can a parent do to reduce their child's exposure, given that this is coming from microplastics, food, dishwasher, rinse aids, so many different places? And he sent in an answer for us, which I'll read, which is, these impacts are really dose dependent. So you have to decrease the doses below toxicity. Parents must get used to reading food packages and looking for those emulsifiers that he described, looking at cosmetics and cleaners. It's important to dilute these much more than we do. And there are certain detergents that can be equally efficient if they are used 4 times or 10 times less in a dilution. He said that instead of packaged food, when you go to the supermarket, decrease those packaged foods that you buy and increase intakes of fruits, vegetables, and healthy options. And a simple hack for microplastics: opening big packages at home inside your house releases a lot of microplastic into your local environment. So this should be done outside of the house, and then the item brought into the house later. And then finally, he said, there are many things that regulatory agencies must do to keep these exposures minimal, and it's really their responsibility rather than the responsibility of individuals. Okay, and we'll continue to answer the questions that are coming in the Q&A. And publishing this so everyone can see them. So it's my pleasure now to introduce Dr. Ian Miles. He is head of the Epithelial Therapeutics Unit at the National Institute of Allergy and Infectious Disease at the NIH. His research focuses on the skin microbiome and its role in the development and treatment of eczema. Dr. Miles is also a board member with Global Parents for Eczema Research, and he'll be talking about advances in the detection of skin barrier changes in early life that will hopefully allow us to intervene with children early on to prevent AD. So thank you very much, Dr. Miles.
Okay, I work for the government. I don't have any disclosures. And I would want to stress what Dr. Actis really highlighted is this mass increase in atopic derm starting around 1960, 1970, really speaking to the fact that something in the environment must be driving this. And we're going to talk today about ways in which we're trying to figure out, trying to tease out what those environments are. So Pretty good explanation of the microbiome, I always feel, is Richard Scarry's Busy Town. So every spot on your body has all these different organisms living on there, and they each have their own separate function. I won't pretend that we have all this ironed out, but for example, the same species might end up having different functions between a tow truck driver or a trombone player. If you were to go to Eczema Town and say, like, all right, well, what would be problematic? We've discussed the barrier functions, cracks in the streets and the buildings not quite fitting together properly. You have itch and inflammation, pretty significant dryness and arid climate, and the normal organisms that are supposed to be living there getting kind of pushed out by the opportunist of Staph aureus. So as discussed, with various therapies, you can restore this back to where it was. You can get the skin back to being healthy and get the microbiome back to being where it belongs. But this doesn't really answer the question of not only, well, why did the skin go down the route of eczema in the first place? And also, why would it go back and forth? Why do we, everything's under good control, but then somehow you get a flare despite not having any obvious trigger? So the first thing that had to be done, because, as you know, this is a pediatric disease, most kids are going to present very early in life. You can't go biopsying and doing a bunch of invasive tests. And this is just me doing it in the break room. So I'm just showing you conceptually what it looks like. These are kind of skin swabs. Obviously, they look way more professional than this in real life. And then I'll show you a picture here of what the tape strips are. It's hard to see, but it's like a very expensive piece of Scotch tape that goes on to the skin. And then this little plunger has a spring load in it, and you can feel it on your thumb. So you kind of standardize the amount of pressure that it goes on. And then, they peel those off and then can send those off for various analyses. So neither one of these are painful. That's the important part. So now we want to look at, like, all right, well, what dictates how the different body sites are supposed to function? The previous lecture talked a lot about the gut, but the gut microbiome is very different from the skin. So the location on the body is by far the number one determinant of whether or not, of what organisms live there and what they're trying to do. The second one is actually the level of industrialization that you live in. So if you are in a highly industrialized country, they can look at your microbiome and predict that, which is more than a little disturbing. And then age, where the younger you are, you have a different microbiome up until about 26, and then things seem to lock into place. And so with these analyses, what we're trying to do is decide, all right, if one of these towns went on to develop eczema, could we predict, was there something, like a fire hydrant that was there before all of the pieces started falling apart that predicts how they were protected? And so these are several studies. This first one is in the stool. And so they collected stool samples and then tracked patients to see who developed atopic derm, allergic rhinitis, asthma, or food allergy, and then the combinations therein. And we're just trying to predict, who went on to develop those different diseases. And the short version of this is they're basically aligning microbiome age versus what your actual age is. So like I said, as you're younger, you have a different microbiome as you grow up. And you can kind of think of it like milestones, right? You should expect your kid to be crawling by 9 months. The problem with this is the first step, okay? So we don't exactly know what it means, that if you look here, the light blue is the healthy people, and then these other lines are the patients with allergic disease. They have a delayed aging of their microbiome. And we don't know exactly yet, okay, well, what does that mean? What would I do with that? That, again, this is the first step, but it's saying that before the disease is even picked up, you can find abnormalities in the gut microbiome, and whether those are emulsifiers or some of those other things, again, they don't yet know. A little bit more detail is known on the skin. So if you were just looking at the different organisms, the microbes on the skin, and say, all right, well, what are they supposed to be doing for you? Really, a lot of it is around ceramides, which Dr. Beck talked about. And this is just a very rudimentary summary of that pathway to sphingomyelins and ceramides, sphingosines, phospholipids, and omega-3s. And I'll show you a more complicated one in a bit, but this is the key one. You can go to the grocery store right now and buy any kind of lotion that brags about having various ceramides in there. And so the organisms, one of their jobs is to help either produce ceramide for you or produce it on your behalf. And so this was mentioned earlier. So they took those tape strips, those little stickers that I showed you early on, and then followed people for 2 years to see who would develop atopic derm. So family history was a risk factor, but what was interesting, a lot of the things that you think of that go along with that did not really bear out. So C-sections and season of birth didn't bear out. Flagrant expression was useless in terms of its ability to predict who went on to get atopic derm. But as was stated, family history, an increase in IL-13, which doesn't really speak to, did the IL-13 come first or did it come after? And then these abnormalities in that ceramides and sphingolipids, those individuals had a 54-fold chance of developing atopic derm. Two other papers are very similar. The first one, again, using the stickers. So they are looking at who went on to develop atopic derm in a different cohort, and they find the same thing. Of all the different metabolites, they look at this imbalance in sphingomyelin and ceramides is the most predictive. Another group took a slightly different approach. They found increased inflammation. They did not confirm the IL-13, so it might just be a difference in their approach. But again, they did show abnormalities in phospholipid metabolism. So again, another study speaking to the same defects. One that we were involved in was using the swabs, and then we're just looking at the microbes of kids followed out to 3 years of age, and again, who went on to get eczema and who didn't. And the upshot of it is these glycosphingolipids, again, those same categories, that there was a defect in the patients well before, this is in their first week of life. So this is well before they've ever developed atopic derm. So we're picking up these same lipid abnormalities, but we're picking them up in the microbiome specifically, that the microbes are not making the correct amount of lipids for you as a way of predicting who would go on to get atopic derm. And so, as I said, the ceramides, this is what makes it difficult when you go to the grocery store and they say, oh, look, we have ceramides in there. This is the full ceramide metabolic pathway. So you can see it's a total mess. It's not really that simple to say, well, let me just, extract one of these and put it into a bottle. But the upshot of what I'd want to stress here is that if your town, right, if your skin town is producing the correct amount of these lipids, then it looks like you're going to be okay. You're not going to go on to develop healthy skin, or it's not going to go on to develop eczema. But now we're looking at 5 papers just this year alone and more, showing that if your skin is not producing the correct amount of those lipids, either directly or maybe through the microbiome, that is who ends up having risk for atopic derm. And so, the reason why this matters is kind of these traditional approaches, all of these anti-inflammatory medications, while they can be incredibly effective, they're kind of stopping a process from moving forward, and they're not really telling you what the first domino to fall is. And why did my kid get eczema in the first place? And then why are they having a flare when you don't perceive any change in anything that you're doing in your life? And really what we want to know is who's knocking over the first domino, right? What is the environmental factor that's doing that? It was talked a lot about in the gut. Our lab works mostly on air pollution as a driver of skin disorders. But these two things combined really then start to give you an idea of, if we can, now that we have non-invasive ways of looking at this and predicting who's going to go on to get eczema, could we start to look for different toxins that influence these specific metabolic pathways? And then could we start doing actual targeted interventions, either microbiome treatments, or, as we'll hear about in the next lecture, various ideas towards barrier repair to happen before. And so hopefully you can, even if that first domino gets knocked, you can stop the process very early on. And so with that, I will open for questions, I guess.
Thank you so much, Dr. Miles, for that excellent presentation. If we could stop the share, we'll do some questions. I think that was fascinating. And one of the things that we wanted to really highlight at this symposium is these are breakthroughs, major breakthroughs in our ability to detect who will develop eczema. And so many of those papers were published just this year. So this is really the cutting edge, and it's opening up the possibility to really be able to predict who will go on to develop eczema and to come up with interventions to prevent that from happening. So it's super exciting. It's a bit technical, but it's a major advancement, I think, in the field. These tests are not invasive. They're not biopsies. They're not painful. And so we can use them with children in a way that's going to be very, very effective. So I think it's extremely exciting. We did have some questions come through. And one of them comes from an audience member that asked, where are these tests available? Or sort of how soon will they be in the doctor's office? And do the tests get to the specific abnormalities that we see with atopic dermatitis? Can they really pinpoint what's going on for that individual?
Yeah, so the problem with the test as it stands right now is really the analysis side of things. I have full confidence that every pediatrician in the country could easily send this off. The problem is getting the results back. I mean, this is either RNA-seq or mass spec, which are not typically used in clinical practice. So they're not there yet in terms of that. It's like a logistics problem, right? The science is ready. Like you could, in theory, start screening children today and send these things off. It's just that they'd only do it from research purposes, not for, something that you could bill an insurance or some kind of central payer. And then in terms of does it get to the heart of it, I would say yes and no. I mean, it clearly is pointing to just consistent one thing after the other. It is a sphingomyelin phospholipid ceramide problem that predates everything that happens. Everything that happens comes subsequent to that. And that really speaks then, okay, well, what are the different environmental toxins that are disrupting that particular lipid pathway? What can we do about it? Could we put, and we'll talk about the barrier the lotion treatments next. But it would make a difference whether you would use a topical that didn't have any of those things to replace it versus one that did, right? So like, I think we are getting there. I don't know. I think you'll probably see treatments outpace the diagnostic test. You'll be more likely to have somebody say, well, why don't you just use this lotion if you're high risk, before you get into developing an actual mechanism to pay for these kind of tests.
Great. Thank you for that answer. And I think because we know it does seem to be a lipid change, a skin lipid alteration, it does point to what interventions might be most appropriate. And that is very helpful. The science is guiding us there. I know that we have one more speaker and a lot of questions coming in the Q&A, but just one more question maybe before we move to our final speaker. Why are these lipid ceramides so important in the development of eczema? And I know that's a bit of a technical question, but could you give us the Cliff Notes kind of answer?
Yeah, it looks like they are necessary to properly stick your skin cells together for that barrier function, and then they interface with the immune system in ways to keep everybody in line, I guess. I mean, they're I don't want to call them anti-inflammatory per se, but they're just there to regulate the immune system. That'd be the best I could give you.
Thank you very much. And there are some questions coming in the Q&A. So if you have a moment to take a look at those and answer live, that would be wonderful. So thank you very much, Dr. Miles, for your excellent presentation. So I'm excited to introduce Dr. Helen Broff, who will be our final speaker today, because she's really going to move us from how do we detect this, how do we understand the changes in the skin barrier that are happening, to what can we really do about it, and what are those best strategies for prevention once we know a child is high risk. Dr. Broff is a consultant in pediatric allergy and head of service at Evelina London Children's Hospital. Her research interests include early intervention and prevention of allergic conditions of childhood. Dr. Broff is leading the multi-center European ProNut study and was co-author of the LEAP, or Learning Early About Peanut study, which was a landmark study. So with that, thank you very much for Dr. Broff for joining us today.
Thank you so much for the kind introduction. So I'm going to be talking today about addressing skin barrier defects early to treat and prevent atopic dermatitis. I work in London. I work at Guy's and St Thomas's, King's and the Evelina London. My disclosures mainly are around the SEAL study, which is a large interventional study looking at treating children with the early signs of eczema to prevent food allergies, which is ongoing in 4 centres in the US and in our centre in the UK. So to start with, eczema and cutaneous sensitization. So I'm going to talk about some of the things that other speakers have spoken very eloquently about, particularly the detergents, Dr. Chesniakis, and then some of the pH issues with the skin by Dr. Beck, and some of the other types of mediators that will break down the skin barrier. So these are all contributing factors in addition to genetic predisposition that then breaks down the skin barrier. And it is a complex interplay. These are some of the mechanisms through which environmental pollutants, detergents, and infections and genetics then lead to an impaired skin barrier function. And then that can lead in itself to food sensitization and allergy, but just with dry skin, because we know that in itself is also a risk factor for food allergy. But this can then lead to skin inflammation. I really like Dr. Beck's comment about the itch that scratches. My daughter also has eczema, and I do see that her skin is fine, but she's scratching it, and then that's when I see the changes on the skin. So we know that microbial disruption of the skin and of the gut is a predisposing factor towards the development of atopic dermatitis. We know that the skin microbiome in children that have eczema is different from in children that have healthy skin or non-eczema skin, and that's very nicely displayed with the previous speaker with the different towns of microbiomes. And you have an abnormal skin barrier and an abnormal microbiome even at rest, which then becomes more abnormal with more Staph aureus, less commensals when there's a skin flare. There's reduced microbial diversity, and I think we've talked about a lot of things that are damaging in terms of pollutants and chemicals and plastics, but also I think there's been a reduction in diversity, both in our gut microbiome and in our diet. And this is, means that what we have is more of the pathogenic strains and less of the friendly strains in the gut. So this, for example, you can see that the Staph aureus can be increased, Clostridium difficile can be also implicated, whereas the good bacteria like Lactobacillus and Bifidobacterium, if they're reduced, can then increase the risk of a development of atopic dermatitis. And this is from fecal samples before the onset of eczema. There's a lot of interest in probiotics for eczema prevention, and from a therapeutic mechanistic understanding, these are many different ways in which you could potentially see probiotics preventing eczema by helping to mature the dendritic cells, increasing T regulatory cells, reducing the inflammatory responses of Th2 and Th17, then directly reducing TSLP expression on the skin, which is an alarmin, and then leads to the downstream signaling of allergic pathways, and then by directly improving barrier function. However, the studies that have looked at actually supplementing probiotics have not been as successful as we would have hoped. And the only guidelines that even give a weak recommendation for probiotics are the World Allergy Organization, and they recommend probiotics in a weak recommendation in pregnant women in their final trimester, breastfeeding women and infants. This is a study that looked at probiotics for the prevention of asthma, and this was published in 2015, and it was shown to favor probiotics. But some of the issues, and this was in pregnant, breastfeeding and infants, the issues is that they looked at lots of different bacterial strains, and the designs of the studies were all very different. Since then, there's been another systematic review and meta-analysis, and this was published in 2019, and they did show that probiotic treatment was associated with a reduced risk of atopic dermatitis, but that you needed to do it both in the prenatal and the postnatal period. And there's a lot of interest in this because the fetus is exposed to the amniotic fluid, which has microbes in it from the mother. And there's a belief that there's some imprinting or some programming that occurs in the fetal gut that allows it then to be more resilient against some of the more Th2 influences, particularly, for example, if the child has a C-section and therefore is not exposed to the microbiome from the birth canal. This may be why just doing them postnatally doesn't work, but only giving them prenatally doesn't work. So you need to have that continued supply of the protective microbiome before and after birth. Skin barrier therapies, again, it would be wonderful if we could just find a nice cheap cream that we could put on the child's skin from birth, and that would solve all our problems, protect the skin barrier against all of those nasty things in the environment, those peanut allergens that then drive peanut allergy. However, unfortunately, looking at some pretty large studies, there was no significant impact of using eczema in children from birth to prevent the development of eczema. This was published in 2021. And then shortly after that, there was another meta-analysis which did a little bit more sort of stratifying and looking in detail. And they found that if they looked at just the high-risk population with preventative emollient therapy, that there was some reduction in atopic dermatitis with a risk ratio of 0.84. And they said also, when they looked at the different studies, looking at whether they assessed the eczema whilst they were still applying the creams or after a gap in applying the creams, that the children who were still having the creams applied had a reduction in atopic dermatitis, which suggests that perhaps this treatment could prevent the development of eczema rather than actually prevent it by delaying it. So it might come later once you stop the creams, but that would provide enough time at least to get some of the food allergens into the diet through oral tolerance induction. But in this study, there was no benefit on food sensitization with preventative emollient therapy. Then I'm going to talk about the Stop AD study, which has happened since then, published in 2013. And this is really quite an amazing study because it goes very much against what the previous meta-analysis showed. So this was done in Ireland, in Cork, with Jonathan Hurahan's group. And there they have a very sort of different way of managing mothers after birth. They are all routinely kept in hospital for 3 days. In England, in London, you're out within 4 hours. And because of that, in Cork, they're able to keep the mothers there for 3 days, and they're able to do a lot more, promoting breastfeeding, etc. But in this particular study, what they were able to do was recruit 321 high-risk infants whose parents had a history of atopy. And they were given a Vino Demexa fast and long-lasting balm, which is a ceramide-containing moisturizer for 8 weeks from birth. And so these children had the emollients applied from in very, very early on in life. Around 3 days of life is when they first started, which is much earlier than all of the other studies because of their particular setup in Cork. And this may also be why they had such a high adherence. So the twice daily application was 73% by 8 weeks, which is much higher than most of the other studies. In the control arm as well, there was very little contamination of the control arm with families applying moisturizers as well. And when they looked at what happened after the 8 weeks to see if they were still putting lots of moisturizers on their child in the intervention group, that wasn't the case. And they were both not really using moisturizers after the 8-week period. And these are the results. They showed that in the children that had the moisturizers applied just for 8 weeks from birth, there was approximately 50% reduction in point prevalence of eczema at 12 months, and about a third reduction in cumulative incidence of eczema at 12 months. Now, there were no differences in skin infections during the 8-week intervention period, and this is in difference to the other study called the BEAT study, where it was much more hands-off, and people were potentially not washing their hands before applying creams onto the child's skin. And there was an increase in skin infections in the BEAT study. There was not an increase in skin infections in this study. And this study happened after the BEAT study, so lessons had already been learnt about ensuring that parents were informed to wash their hands before applying creams. And then going back to the trilipid story, which is the ceramide story, so there's quite a lot of data, which I won't present now, which shows that trilipid creams can improve the skin barrier. And this is a study pilot study in Australia showing that in children that had trilipid creams that were applied from around the time of birth, these were high-risk infants, that there was a not significant improvement in visible eczema at 12 months, so 16% in the control arm versus 5% in the intervention arm. But what they found, which was really interesting, was that they were able to prevent sensitization to foods at 12 months of age if the families were applying the creams in the way that was recommended. i.e. the protocol group. There was 0% sensitization to milk, egg, or peanut at 12 months. So to summarize, the petrolatum-based creams may delay eczema, may prevent eczema in high-risk children. There may be an impact at the time of cessation of treatment and assessment of AD on this. Then the Stop AD trial sort of goes against that. And then there potentially is an increase in skin infections if the right advice is not provided about washing hands before applying creams. This was found in the BEAT study, but not found in the Stop AD study. Lipid-based creams showed a 50% reduction in point prevalence of atopic dermatitis. And the PEBBLE study also showed positive trends for atopic dermatitis prevention. And there's a much larger study being carried out in Australia, and we are awaiting the results of this. This brings me to the SEAL study, which is an NIAID-funded multi-PI study in Stanford, National Jewish, Chicago, our site, and newly Harvard. We're looking at whether children between the ages of 0 to 12 weeks of age can have food allergy prevented and, of course, eczema treated and prevented from having flare-ups with proactive skincare. We're using 2 different types of emollients, Aveeno versus Acrylipid, and we're using proactive topical steroids, which means using topical steroids twice weekly after you've maintained the flare of the eczema for 16 weeks to see if this can prevent transcutaneous allergen sensitization and the development of food allergy. And this is ongoing. We've recruited approximately 200 participants. Future directions. So for the skin barrier repair, I've talked about moisturizers. I've talked about proactive therapy with topical corticosteroids. There are many other therapies which I didn't have time to go into today: filaggrin replacement, filaggrin enhancement, JAK inhibitors, AHR agonists. So there's a lot going on. And then there's a lot working in the skin and the gut microbiome as well. So my take-home messages. Probiotics used during pregnancy and in the postnatal period may reduce the risk of developing atopic dermatitis. But the issues with different study designs and different species all being lumped together is making it difficult to interpret, and so more data is required. Emollient use in infancy may prevent eczema in children who are atopic, but not in children who have non-atopic parents. I always have a real plug here about petrolatum-based moisturizers and any moisturizer, really, to make sure that the families are not inadvertently putting bacteria on the skin by not washing their hands before applying creams or by putting their hands inside a pot of emollients. So ointments, for example, usually come in these big pots, and then people put their hands inside, apply it on the child's skin again, put their hand inside again. And there's good data to show that they're contaminated with bacteria. We know that bacteria then make the skin more inflamed and increase the risk of developing food allergy. The Stop AD trial showed that early ceramide emollient use in infancy may prevent eczema and food sensitization. But actually getting that into clinical practice in the way that they did would be very hard, because you'd need to have a setup like they did. They actually stopped the study after they saw the BEAT study findings, because they thought it was not worth continuing to follow up the families to see if they were preventing food allergy. But then, since they had these dramatically positive findings, they now have funding to assess these children at 4 and 5 years of age for food allergy. So we are awaiting this data. These are my acknowledgements. I'd like to thank the whole team at KCL and all the different people I'm working with internationally. And thank you for your attention.
Thank you, Dr. Broff. Excellent presentation. I think one of the things that I take away from your summary is that these strategies for how to prevent eczema and food allergy need to be very nuanced in how we do them. The method that you choose, the timing is important, when in childhood, the exact type of intervention that's used, what probiotic, what emollient, these things all matter. And I appreciated how the latest research is really shedding light on those details to help us pinpoint what's really going to work. So our first presentation talked about the changes in the skin barrier that occur in atopic dermatitis and what we now know about that. We talked about how that skin barrier becomes damaged through Dr. Actis's presentation. Dr. Miles talked about new breakthroughs which are allowing us to detect those changes and to know that eczema may actually develop. And Dr. Broff's presentation is starting to point to solutions. So It's been a great lineup. We do have some questions coming in the Q&A, and I think one that I wanted to ask you, Dr. Broff, today was, given what we now know, what are the critical unanswered questions that need to be resolved in order for prevention strategies to become adopted and for us to really be able to ward off eczema in early childhood?
I think, to be honest, getting the basics right is where we start. So not inadvertently causing more harm by putting bacteria on the skin, by not advising people to wash their hands before applying creams, by giving them pots which they put their hand into, really basic things like that. There's so much misinformation about eczema. When the parents see that their child first has eczema, the child probably only has eczema, which is mild to start with. Then they're given all the wrong advice, and the eczema gets worse and worse and worse and worse. And then they come and see a specialist, but by that time the child has already had eczema for about 2 or 3 months, and some of the damage is already done. I think free online educational resources are incredibly important. We have to make this accessible to all people. There's a wonderful, another time I'll present that. There's many in all over the world, but eczemacareonline.org.uk is a wonderful, designed, free from industry, created by parents and by adults that have eczema. It just gets all the messages across properly. People are really scared of using topical steroids, so they don't use them, and then they get, really terrible eczema. Then their child has to have oral antibiotics, which then damages the gut microbiome further. So I think before we start going into JAK inhibitors and all this kind of stuff, we just need to get the basics right, so that when a family gets in with a mild eczema, that they, get access to the right information, they can manage their child's skin really well, really quickly, not, put creams on their skin that have peanut oil or sesame oil that could potentially lead to the development of food allergy. It just get the basics right. I think that's really where I'm at, because I see so many families with I feel so bad for them. Their babies are so badly affected with eczema. And if only they'd been given the right advice, several months ago, that the whole thing would have changed.
Wonderful. Thanks for that answer. And we'd love to share that resource that you just mentioned with our attendees today. So we are now at time. Thank you so much for attending this year's symposium. And thank you, Dr. Broff, for your excellent presentation. Please join us next year when we'll be looking at the link between the gut microbiome and eczema, a very exciting topic. And if you'd like to receive an invite for that symposium, please sign up for our newsletter, which we'll put in the chat in just a moment. We also invite you to share your reflections about what you learned today on social media or with us directly at info@parentsforeczemaresearch.com. We'd love to hear what you thought. And so with that, thank you everybody for joining us for this year's symposium, and have a wonderful rest of your day.